Bio 30. Today's class is about gene mapping, which is looking at how we determine an approximate location of genes on a chromosome. So the way we can work on this is by studying linked genes, and linked genes would be ones found on the same chromosome. And so the idea here is that we know that if they're on the same chromosome, they should all end up in the same gamete, because if we get an individual, a male, for example, who has these two chromosome number 10s from his parents, his gametes will either have this one or this one in that gamete. And so the possibility exists, though, that crossing over may occur. And so it's actually this crossing over part that is the element that gives us the ability to do gene mapping and tell where the genes are. And if you can recall, when in prophase 1 of meiosis, the two number 10s come close, the inner chromosomes could swap some genes, and they would be genes of the same thing. So where they had like this chromosome had a dominant and this had a recessive, his parents had different genes for the letter B, they could swap places here, and the chromosome then will have a combination of genes, not just what we started off with from the one parent. So I'm going to explain how this works. This is an example of how they gene mapped chromosome 5 in wheat, and so they know where all these genes sort of are placed. And the idea there is that in knowing that, if they want to do gene modification and take something out and maybe replace it with something else, some grains, for example, are genetically modified to carry vitamins or some resistances to some chemicals or whatever they're modifying it for, maybe for flavor, maybe so it doesn't freeze. Anyways, they like to know where on a chromosome these genes are located. So when we talk about crossing over, that's kind of the key aspect of this. And I like to just have you visualize having a string of 20 friends that are running through K-Days, and you're headed to the concert. The ones that are, if you are right next to your buddy and everybody's holding hands and running through, it's most likely that you'll end up 15 minutes later still with your buddy. But somewhere along that string of 20 people, the line may break as you're weaving through the crowd and running. So the idea is the further apart you are, like first guy and last guy, it's more likely that you will be split up and not end up together. So the idea is the further apart those two genes are on a chromosome, the more likely they are to be split up by crossing over somewhere within that chromosome. And so that's how it comes to the concept of gene mapping, is that when they look at when they look at the results of crossing two individuals, and they see results that they didn't expect because those genes should not have been in that combination together, that means crossing over happened. And the amount of times they see the two genes that they're studying having unexpected results would then be considered a clue as to how far apart they are. So if they very often are found to have crossed over and be in different combinations, like 10% of the time, they would consider those two genes then being 10 units apart on the chromosome. But if there's only like 1% of the time that they ever are separated and crossed over, that means that they're very close together. And if it's 1% of the time, they would be considered to be one map unit apart. So in this example, they're looking at genes for this fly, and they're saying that for the, for example, if we're looking at wings and legs, they know where they are located on the map based on how often they are in different, how often when they cross, looking at those genes, how often they are in different combinations than what they expected. So I'll show you this example with blood types and this nail syndrome. The nail syndrome is a dominant condition. The blood type B is more dominant than O, so you can be a type B if you're carrying a B and an O. And you'd be type O if you have OO. You'd also be B if you have BB. But what we're looking at here is this, I'll just highlight here, this male, he's got type O on both of his chromosome 2s, let's say they're 2s, and he's got O and normal nails. So no matter what, his gamete will always have type O and normal nails because that's the only combination that he has on both of his chromosome 2s that both of his parents gave him. She, though, has type B blood with the nail syndrome on one chromosome and O and normal nails on the other. So her gametes can carry one or the other of those chromosome 2s. So the idea is that if their kid ends up with this one and this one, this is what their child would be, nail syndrome and type B blood. If their kid ends up with these two, they're going to be normal nails and type O blood. And that would be the only two kinds of possible kids they could have for those two conditions, because that's all the gametes that are possible. However, scenario 2 says, but if there's crossing over, first of all, it wouldn't matter if his crossed over because he's only got the same on both. So he's always going to have this as his gametes, type O and normal nails. However, with her, if her two chromosome 2s do cross over in prophase of meiosis, then she could have different combinations. And so her combinations could end up being different than what they were if they hadn't crossed over. And so if crossing over does happen, then it shows that she could give different combinations when joined with this guy. The guy still has only one type of possibility of a gene combination in his sperm, but in her eggs she could have the scenario where she never crossed over, or she could have the scenario of where she did cross over. And so when we look at what, like if they did have a test group of 50 kids and we looked at their kids, we would see that there was the majority of the time, 45%, majority of the time, what we expected to have as their kids. So we expected this to happen, as you see over here. However, 5% of the time we got something unexpected with normal and B blood, and also 5% we got the nail syndrome and O blood. We did not expect to get these two. And so that's what I mean by unexpected results. The unexpected results are showing us that 10% of the time, because this is 90% of the time, 90% was our expected results, the same as this, but 10% of the time here, 5 and 5 is 10, 10% of the time we got unexpected combination because of crossing over. Now, because it's 10% of the time, it is simply going to be known that these two genes, the blood type and nail syndrome, are 10 units apart, 10 map units apart on chromosome 2, if this was chromosome 2. Okay, so the amount of times that testing with them, the percentage of times we get recombinations, is what we will say is the distance they are on that chromosome, how many map units apart. So 10% of the time they are found to have change their like their location, and that's because they're 10 map units apart. And we'll see how we do the examples on this, and it's not a very hard thing to do. We can just look at things like if gene, or if they have a set of data that says eye and body color, they say recombinations 4% of the time, and in body color that we're studying in these flies and wing type, if they do crosses looking at just those two genes, then 6% of the time there appear to be recombinations. This kind of data is enough to draw a map, gene map of where they're located. And basically, what you can also see here is that the other part I meant to write down is that eye and wing, if you study those, they are 10% of the time they are found to be recombined. So this is a kind of a data that would be given, and then you can draw a gene map like this by saying which is the furthest apart here. 10% of the time it's eye and wing. So I would draw a line that's your chromosome and say that eye and wing are 10 units apart. They are my furthest, so you take the furthest and draw them first. And then I always draw my next furthest next, and its body and wing is 6. So I go to the wing and say the whole thing is 10, so 6 is about over here, and that's where I'm going to draw my body, and 6 units apart is body and wing. I go back to the last one, and eye and body are 4 units apart, and that totally matches up with the fact that the wing and eye is 10. So that is as easy as it is. We're just going to take a look at how we do a few more examples. Now, when we were doing dihybrid crosses, this isn't an exactly great picture, but we were looking at, first of all, what 2 chromosome 6s had and what 2 chromosome 7 had, and that's what we were doing for dihybrid crosses. So that's when we were doing crosses like that parent 1 had here little a, big B, parent 2 had big A, big B, and we were crossing like that. With linked genes, we were just looking at crossing these 2 people and checking what he had on his 2 chromosomes, but the genes were on the same chromosome, okay? And what she had on her chromosome 12s, but remember, we're looking at 2 genes that are on the same chromosome when we cross them. You guys don't have to do crosses with linked genes in our curriculum now. I just wanted you to know that when they test these crossovers, they are chromosomes that have we're looking at with the genes on that same chromosome. And all you need to do is look at the, I'm skipping back past this because you just have to look at the data that's given. So here's an example where they just say there's genes A, B, and C. Let's draw the gene map. And you're going to take a look at the biggest number first and say that A, I'll just redraw it for you, A and B, I know is 12 units apart. My next biggest number is that B and C are 7. So you're just saying B and C are 7. You're going to just eyeball that in. Okay, you're just saying B and C are 7. And A and C are 5. And of course that matches up to 12. And so you know that you've done a proper gene map. And so a question may ask you, starting at A, what's the order of genes? And your answer would be A, C, B. If they say starting at B, what's the order? You'd say B, C, and then A. I want to say that some people may draw this exact thing the opposite way and have drawn it this way. And I just want to tell you that that is not incorrect. You can actually hold a pencil one way or flip it and hold it the other way. It's still the same pencil. So this is This is just as correct if you drew your gene map opposite to the ones I will show you in my answer key. If you drew it flipped, that's still correct because you're just looking at what the order of those genes were. Now, when I have students draw these, I always say, you know, this is kind of like looking like a working copy. Always end up with a nice, I'll just start over with my nice diagram here. That doesn't want to erase very well because I put the wrong thing. Okay, so if I was to do this example, I would end up, I'm just drawing this again, to say that when you finally, you know, get your working copy figured out and know what your gene map looks like, I would do a final gene map that looks like this. And what that is where I've put my genes, usually on the top, or definitely on one side of your chromosome, and put your distances between all genes below. So even if sometimes they don't have all of the information about every single distance between all the genes, your gene map should. And sometimes it'll be more than 3 genes, sometimes it'll be like an extra gene in there, and they've given you enough information to draw the map, but you still have to fill in what distance each of the genes are between each other. And so we'll see as we get, for example, this one has 4 genes, and it's showing you the gene map, the recombinations in a grid format. So how we tackle this is, first of all, realizing that this is just the same as this information, except they've put it in a grid and they're showing it twice. So remember I said we look for the biggest number, and you see a 14% here between F and E. We know it's 14 map units apart, so that's my biggest distance. And you might say, wait a minute, there's another 14. But I'm going to say to you that it's still telling you the same thing, that E and F are 14 apart. They just have it written in 2 spots in the grid way. So I'm going to draw my chromosome. I'm going to draw, it doesn't even matter if you draw E and F the other way apart, your gene map will just be flipped of this one. Okay, so we know that E and F are 14 units apart, and so we're next going to go to the next biggest number that we see. I mean, we do see it twice, but it's saying that G and F are 12 apart. So I'm going to go from F and say if this whole thing is 14, but G is 12 apart here, I know where to kind of eyeball it in. My next biggest number is the 10, and that's E and D. So we know that the whole thing is 14. E and D are 10 apart. That gives you a spot you can eyeball that one in. And in fact, we now have placed D, E, F, and G on my map, and that is your map. But what I was saying earlier is that you want to have distances between all of them. When they show you this grid, if you're looking at E and G here, you just have to go to your map and say E and G, it says 2. Does that make sense? G and D, it says 8. And D and F, it says 4. So let's see if those being put in there actually add up to 14. And they do. So if I was to do a final gene map of that example, I could just say I will put my 4 genes neatly on my chromosome, put my distances below, and this would be considered a complete and final photo of the gene map. Okay, so the distances, the genes are on top in this case, and the distances are on the bottom, and it's all nice and neat. Over here, this is kind of our working copy where we were figuring out the distances. What I meant by having to fill them all in is that sometimes all of the information is not there. So they may have given you the information to determine the 8 and the 4, and you might have had to say my whole thing that is 14 minus 12 is how I figure out my 2. So sometimes they give you one that's not figured out and you just have to subtract. problem 3, here you can see that the 24 is your, oh, by the way, for this one, if you put your E and F in opposite locations, your gene map should be an exactly flipped one of this one. So you can not worry if yours is exactly opposite. It depends which way you put your, in this case, your X and your W, which are the ones that are the furthest apart. And depending on where you put them, is how your map is going to look. So I see W on this side, so I just randomly I'll pick W on this side, X on this side. And I know they're 24 apart. And then looking at this one, I'm going to notice that my next biggest number, Y and X, sorry, Y and Z, are both ones that are floating in the middle somewhere. So we can't really use that second biggest number in this case because we can't see where it sits next to the X and the W. So for this one, you're going to go to the next lowest number in this case, which is the 7, and that's the Y and X. And seeing that Y and X are 7 and this whole thing is 24, I might put the Y about here and say this is my 7. And then you can go to the next one, which is a 5, Z and W. And so you know that your Z is probably over here. It's 5 apart from the W. And now you can use the 12, that's the Z and the Y, because you know that the 12 goes here. And we still have a V that's 2, and so we know the 2 is over here. So my V is 2, this one is 5, this one is 12, this one is 5. And the final gene map here, basically, we just clean it up a little bit, and I can just keep this one. But this is our final gene map for these 5 genes on this chromosome. Okay, so this is just the answers of the same ones that I just did. And so we are going to wrap up this lesson, and I'd like you to try some gene mapping. Remember the chart of percentages is just telling you how often crossing over happens so that you know how far apart those genes are on the chromosome, because how often they recombine, the percentage of the time they recombine when crossing is done with those individuals, looking at only those traits, 2 at a time. So we're only looking at when they got the 12%, they looked at only A and B when they did the crosses and looked at the offspring and said 12% of the time we had combinations that we did not expect to see. So 12% of the time crossing over must have happened. That means that those 2 genes A and B are